And the bond that hold these together would be the phosphodiester bonds, which are stable and relatively strong. And basically, heat cannot break these phosphodiester bonds. But what can break these bonds would be restriction enzymes. And these restriction enzymes are very important when we're talking about constructing recombinant DNA molecule. So if we have a DNA molecule here and we want to cut it, remove a portion and put in another portion, restriction enzymes, which are considered molecular scissors, those are the ones that are capable of breaking the phosphodiester bonds. All right, is that clear? So bear in mind that you have, go ahead, Shan.
So just out of curiosity, sir, how you actually split one DNA?
When you say split it, what do you mean? Open it so that you have two single strands?
Yes, sir. How you actually cut it, sir?
Okay. So let's imagine this to be the DNA molecule. This is a simplified version, right? And of course, these are the bases that are holding the two strands together, and these bases are held together by hydrogen bonds. So what you have here would be hydrogen bonds. Remember that in the case of A and T, these are double bonds, and in the case of G and C, these are triple bonds. These are hydrogen bonds. So I said that by heating a DNA molecule, it would cause these bonds to break. So it would break these bonds and create two single-stranded DNA molecule with heat.
Oh, sir, I'm actually really think like, I don't know, you may use a laser or something to cut it, sir.
No. Heat alone. You just heat up your sample that has a DNA molecule, and that heat will rupture the hydrogen bonds and cause your DNA molecule to become single-stranded. All right, so with heat only.
Oh, okay then, thanks, sir.
Yeah, so I said that heat cannot break these phosphodiester bonds that are here. All right, so these phosphodiester bonds that are in this backbone, heat cannot break those. So to rupture these bonds, for example, here, or to break this bond here, you need restriction enzymes. All right, and restriction enzymes have that. You have different types of restriction enzymes. So you have certain restriction enzymes that are capable of identifying where there's an A and a G on that DNA molecule, and it will go and cut right between that. You have other restriction enzymes that will recognize where there's a C and a G, and they'll go and cut between that. Okay, so restriction enzymes are extremely important in terms of constructing recombinant DNA molecule, because when you use them to cut a DNA molecule, then you will probably add a different piece in there and join it with ligase enzyme. Okay, so restriction enzymes, those are the ones that are used to cut phosphodiester bonds in that sugar phosphate backbone. Okay, so just recall that in this normal DNA, the structure that you're seeing here, it is actually double helix, right? And it is somewhat twisted in a right-handed position clockwise. Think of like, for example, a rubber band. Even though a rubber band is circular, and this we're talking about here is linear. But you can take that rubber band and you can twist it clockwise, all right? And you'll find that it will become into a smaller, it will occupy a smaller volume, all right? So that is one of the reasons why this long DNA molecule can be packaged in the nucleus in the case of eukaryotes, right? So normal DNA has a right-handed twist to it. Let's look at other DNA structures. DNA can exist in different forms depending on the conditions. We're talking about the conditions within the cells, right? So when humidity is relatively high, DNA is in what is called the B form. And this is the form that was proposed by Watson and Crick. That is the figure that you see right there. This is the form, so that is B form. So take note that the B form has a right-handed or a clockwise helix. All right, so the B form is the normal form of DNA that was proposed by Watson and Crick. When the humidity is relatively low, DNA is found in the A form. So both of these DNA forms involve right-handed helices. In A DNA, the base pairs are tilted and pulled away from the axis of the double helix. So A form and B form has something in common, and that is because both of them are right-handed helices, while the difference between A form and B form is that B form runs directly down a central axis, while the A form is somewhat tilted away from that central axis. Okay? So that's the difference there. In B DNA, the helix axis passes through the base pairs, which are themselves oriented perpendicular to that axis. Okay? Another form, which is the most intriguing, is the Z DNA. And because of its totally unexpected structure, and because of the evidence of its rare existence in cells, zDNA has a left-handed helix and has a zigzag sugar phosphate backbone. So it doesn't run perpendicular to that central axis, but rather it has a zigzag pattern around that central axis. Some researchers even deny or question whether zDNA exists within cells. All right, so that is something that is still debatable, whether that form actually exists. All right. So we said that B DNA is the form typically found in cells. So this diagram here shows three forms of DNA. So we said that A DNA and B DNA, both of them have something in common, and they have right-handed twists, right? The difference between these two is that you notice that here, the B DNA, the two strands run somewhat perpendicular to a central axis, while this one is somewhat, A DNA is somewhat tilted away from a central axis. So it doesn't follow that pattern of being perpendicular to a central axis. The Z DNA, on the other hand, has a left-handed helix. All right, so instead of twisting this way clockwise, it is twisted anti-clockwise. And of course, it has a zigzag pattern around that central axis, okay? So these are the three forms of DNA. And of course, the normal form would be the B DNA, all right? That is the form proposed by Watson and Crick in terms of its overall shape, okay? Structural characteristics of eukaryotic chromosomes. Right? Structural characteristics of eukaryotic chromosomes. It differs substantially from prokaryotic chromosomes. Please bear in mind that prokaryotes have how many chromosomes? Anybody recall? Prokaryotes have how many chromosomes? Nobody recall. Prokaryotes have one single circular chromosome. Okay. So prokaryotes have one single chromosome. And when you compare that to, let's say, human beings, we have a total of 46 chromosomes. So you can see whereby eukaryotic chromosomes are way more complex, because we have to deal with 46 chromosomes, the structure of these chromosomes, how these chromosomes are packaged within that nucleus. While in the case of prokaryotes, that one single circular chromosome is found floating in the cytoplasm in a region called the nucleoid. All right, so you can see the complexity there in terms of eukaryotic chromosomes having to fit all these, in the case of human beings, 46 chromosomes within the nucleus of that cell. So the eukaryotic chromosome complement, the fundamental difference between prokaryotic and eukaryotic is that prokaryotic have one chromosome, while most eukaryotes have a diploid number of chromosomes within almost all somatic cells. Karyotype. What is a karyotype? Have you heard that term before? If so, what is it? Has anybody heard the term karyotype before?
A set of chromosomes.
Say that again, Madison.
A set of chromosomes.
The state of chromosomes. Anybody else?
All metaphase chromosomes.
All metaphase chromosomes. Anybody else?
Sir, isn't it the number of appearance of chromosomes in a nucleus?
The number of appearance of chromosomes in a nucleus. Anybody else? All right. So basically, all what you guys said is correct. Complete set of all the metaphase chromosomes in a cell, and that is what is called a karyotype. So basically, a karyotype is a diagram showing these chromosomes that were in the metaphase stage. So how do you actually get a karyotype to visualize these chromosomes? Well, you have to stain the chromosomes, and you can do so using several techniques. Right, where there's something called quinacridone mustard. You can stain your chromosomes. You just drop it on the cells. It will stain the chromosomes, and that will produce what is called Q bands. So it will produce certain bands on your chromosome. So if you have a chromosome like this, for example, all right, the staining will produce certain bands on your chromosome, allowing you to visualize this chromosome. So from these bands, you'll be able to determine the size of the chromosome, where the centromere is, all right, the P arm, the Q arm, and so on, okay? So staining your chromosome allows you to visualize your chromosomes. There's another technique which is staining for treating your chromosomes with trypsin and then staining it with Giemsa stain. That would produce what is called G bands, which would have a similar pattern as the Q bands. The difference is that for the G bands, you do not need a fluorescent microscope to view the bands. You can view it through a regular light microscope. However, with the Q bands, that requires a fluorescent microscope to visualize it, okay? All right, so several techniques exist that allow you to stain the chromosomes and then allow you to visualize the chromosomes in that diagram that you'll call the karyotype, right? So for most organisms, all cells have the same karyotype. However, the karyotype is species specific. What does that mean? The karyotype of a human being will show 46 chromosomes. Of course, the karyotype for a different animal or a plant or someone would show differently because they have different number of chromosomes, and so it is species specific. All right, so a wide range of number and size and shape of metaphase chromosomes is seen among eukaryotic organisms. So a knowledge of the size, overall morphology, and banding patterns of chromosomes permit geneticists to identify certain chromosome aberrations. This word aberrations means abnormalities. So it allows geneticists to determine or to identify certain chromosome abnormalities that correlate with congenital abnormalities or dysfunction. Congenital means that you're born with it, all right? So one example that you see here on the right is a Down syndrome karyotype. We all know that Down syndrome is a case whereby there is an extra chromosome at number 21. Excuse me. So there's an extra chromosome at number 21, which is an indication that that person has Down syndrome, right? Excuse me. That is congenital. That person was born with that. So, karyotype allows us to detect or identify genomic mutations. Genomic mutations is when you have a change in the entire genome in terms of the number of chromosomes. So in this particular case, there's an extra chromosome. All right, so this would be 2n plus 1. If there's a case of 2n plus 2, 2n plus 3, karyotypes would be able to pick that up. If there's a case of 2n minus 1, 2n minus 2, and so on, karyotyping would be able to pick it up as well. All right? So karyotype would be able to pick up aneuploidies. Remember the term aneuploidies? Aneuploidies is an imbalance in the number of chromosomes. So we're talking about cells that have more chromosomes than the normal or less chromosome than normal. One thing about karyotyping is that it can be done even before someone is born. So you have a woman who is pregnant, probably 4 or 5 months pregnant. They can do what is called an amniocentesis, whereby they take out some of the amniotic fluid. And from that amniotic fluid, they can actually do the karyotyping. So even at 4 months into the pregnancy, that woman can know whether her child will have some kind of major genetic abnormalities. All right, we're talking the aneuploidies. So we're talking about Down syndrome, for example, which is having that extra chromosome at number 21. We're talking about Edwards syndrome, having an extra chromosome at number 18. All right, Edwards syndrome, an extra chromosome at number 18. Patau's syndrome, having that extra chromosome at number 13. Patau's syndrome. All these chromosomal aberrations, karyotyping can pick up. However, there are some mutations that karyotyping would not be able to pick up, such as gene mutation. When you have a mutation occurring within a gene, like for example, a point mutation. A point mutation is nothing more than a change in a single base pair. So when you have a change in one single base pair, do you all know that sickle cell anemia is caused by a change in one single base pair? And when I'm talking about base pair, we're talking about, all right, A and join with T, C, and G. So we're looking at A pairs with T, but if there's a mistake and instead of AT you have GC, or you have it the other way around, instead of A and T you have T and A, that leads to sickle cell anemia. So that is a point mutation. Certain conditions like that, karyotyping cannot detect. All right, so karyotyping can only detect genomic mutations, all right, such as the aneuploidies, where you have changes in the number of chromosomes, and of course, certain structural changes to the chromosomes. For example, deletion. So if a large portion of a chromosome is deleted, karyotyping would be able to pick that up. All right. Yes, this is caused by error in cell division, such as chromosome non-disjunction. So cases like Down syndrome that we have here, Edwards syndrome, Patau syndrome, these are all autosomal, and this results from non-disjunction. If you can recall, what is non-disjunction? It is the failure of chromosomes to separate during cell division. So when you have failure of chromosomes to separate, then it leads to these aneuploidies. Okay? In a human karyotype, the chromosomes are numbered for easy identification. So the largest pair of homologous chromosome is designated as 1, the next largest 2, etc. So you have chromosome from number 1 all the way to number 22. Pair number 22, those are called autosomes. And then pair number 23 would be the sex chromosome. So if you look at this karyotype, you'll be able to discern that this is a karyotype from a female, because when you look at the sex chromosome here, you see 2 X's, and this is pair number 23. So pair number 23 is designated for the sex chromosomes, while chromosomes 1 to 22 are the autosomes that have absolutely nothing to do with the sex of the organism. Okay? All right, so it says the sex chromosomes constitute pair number 23, even though in humans they don't fit properly in the size scale. The X chromosome is a large metacentric chromosome. Do you all know what metacentric means? Did we go through the different types of chromosome based on the position of the centromere? Yes or no?
Sir, isn't metacentric where is the centromere is in the center of the chromosome?
Yes, that's correct. And what Jeren says is correct as well, right? So metacentric chromosome has a centromere directly in the middle. And then you have submetacentric. It has it a little bit on one end. All right. And then you have acrocentric, which has a centromere almost at the end. And then you have telocentric chromosome, whereby it is way at the end and one arm of the chromosome is actually non-existent. All right. So you have metacentric, submetacentric, acrocentric, and telocentric. The Y chromosome is a much smaller acrocentric chromosome, right? The Y chromosome. So bear in mind, I think I said this in a previous class, that because the X chromosome is much larger than the Y, then the X would have more genes than the Y, and so there would be more X-linked traits than the Y chromosome. Go ahead, Shian.
Sir, I confused now because I just think back to the quiz just now, sir. Sir, one of your questions said a male human being has 22 pairs of homologous chromosomes and 2 non-pair homologous sex chromosomes, sir. And over here you have 23, like the human have consists of 23 pairs of chromosomes, right, sir?
Mm-hmm.
So the answer never would have been false, sir, because then 22 plus 2 are obviously 24, right?
What exactly did the question say?
Sir, the male human being has 22 pairs of homologous chromosomes and 2 non-homologous sex chromosomes.
Mm-hmm. Okay, let me see if I can answer you with this next slide.
Okay, sir.
Look at the karyotype to the left, where it says a normal male. So if I can recall, I think the statement says that a normal male human being has 22 pairs of homologous chromosomes, which are the autosomes, as seen right here. It is within this black line. So from 1 to 22, those are homologous chromosomes. But are the sex chromosomes homologous? They are not, because one is X and one is Y. Take note of them right here. So a male would have non-homologous chromosomes. So if I can recall, I think the statement says a normal male, human male, has 22 pairs of homologous chromosomes and 1 pair of non-homologous chromosomes.
2 pairs of non-homologous chromosomes, sir.
It says 2 pairs.
Yes, sir. It says 2 pairs.
A male, okay, Princella is giving me the full thing right now. It says one pair. Jaren said it says 1 pair.
Sir, it says 2 pairs, sir.
Pairs of homologous chromosomes and two non-homologous. It says and 2 non-homologous sex chromosomes. Where does it say 2 pairs, Shan?
What a question. Sir, you can write. Sir, it says 2 non-homologous, and we mistake that for 2 pairs. Because the 2 non-homologous is 1 pair. Exactly.
So it doesn't say 2 pairs.
Sir, that was the question mix you up, sir. Sorry, sir.
Okay. So that question, this karyotype that you see on the left is basically what that question was all about. So 22 pairs of homologous chromosomes, which are the autosomes. And in the case of a normal male, it would have 2 non-homologous sex chromosomes, which is the X and the Y. While in the case of a female, their sex chromosome would be homologous because it is 2 X's, right? As seen here on the right, all right? So this is a normal female. So it has a normal 22 pairs of autosomes, and it has 1 pair of sex X chromosomes, which are the sex chromosomes, all right? So on the left is a human male karyotype, and indications are that it is a normal male. And then on the right, it's a human female karyotype, and everything indicates that it is a normal female, okay? All right, so guys, it is up to this point where we are going to have quiz number 3. So this is posted already on Moodle. All right, I posted this on Moodle yesterday. So the quiz will be on that file that says GE3, which starts from genetic linkage, I think. So genetic linkage and up to this point. Okay. Any questions? Let me give you a little hint.